Good morning, everybody. Let me just get connected here and get situated. Thank you for joining us. My name is Sue Tappan, and I'm joined by Aidan Sullivan.
Good morning.
And we are here today to talk about Neurolucida 360 and our goals for the coming year, 2021. I'm pretty excited to put 2020 behind me and get started on this brand new year. So I'm going to share my screen, and then we will get started. So as I said, my name is Dr. Sue Tappan. I'm the product manager and scientific director for MBF Bioscience. So I'm the product director of Neurolucida 360. So that means that I have a hand in helping to determine what aspects and directions Neurolucida 360 goes currently and in the future. And one of the things that I'm hoping to spend a little bit of time with you guys today is talking about where Neurolucida 360 is right now, and some of the things that we've been thinking of as directions to take it in the future. And what I'd really like is to start a conversation with everybody on the line today about where you think we should devote our energy, because we want to make a product that serves your needs and gets the most benefit for the most users as we can possibly manage. As I said, I'm joined by Aidan Sullivan. Aidan, do you want to give a shout out?
Good morning. My name is Aidan Sullivan, and I work with Sue in the MBF Labs team and do a lot of demonstrations for Neurolucida 360 and involved in the product development process as well. So it's great to be here today. Thank you so much for joining us.
And additionally, on the line, we have one of our senior software engineers, Doug Hoppus, who is on the line in case we have any tricky questions that neither Aidan or I are able to address with specific requests to the software code and development perspective, so that we have an opportunity to have a real in-depth conversation about the topics that you'd like to discuss. Before we get started into the discussion of Neurolucida 360, fair science, and open science, I want to start by telling you a little sandwich story. So I've got this delicious sandwich story I want to share with you. And early in the pandemic, there was this huge run on flour and yeast. Everyone and their brother was planning on baking bread. Sure, they previously brought their bread for their sandwiches, whether it was Wonder Bread or their favorite artisan boulet, but now things were different. Of all the people who caught the bread making bug, there's some who had the ingredients but never got around to it. Others did bake bread but ended up documenting their food fails on Instagram, me, for example. Some had success and also documented their successes on Instagram, making bread until something new captured their focus. You know, for example, whipped coffee. Others really took to it and now have incorporated it into their daily life. They no longer buy bread, and they make all of their sandwich bread. There are people, however, who enjoyed the process of making bread exactly as they desired and enjoyed learning the new skill, but in the end, they just want to get back to eating their sandwich and showing their friends what an awesome, delicious sandwich they've created. So they'll save their new skill for a special occasion when homemade bread is needed. Not always looking for that 27-hour labor of love, not having to spend time wondering if the yeast is active or proofing the dough. They want that fabulous sandwich in their belly, and they may want to share that sandwich recipe with their friends. So back to the store they go, and they choose their price point and quality of bread depending on their budget and their sandwich or dietary needs. I think open source software is like baking bread. It has to be tweaked to your experimental parameters. You've got to understand the components, how and when they should be used, and your recipe matters. You may not just be able to use someone else's recipe with your ingredients, and it will probably take some time and experimentation to get it all figured out. So my question to you is, do you write code in your own lab?
So presented on the screen here is a poll, and you can vote for the option that applies most to you. It looks like we have some responses coming in, so it looks like people are figuring that out. Thank you guys for your participation.
So commercially produced software is like getting that loaf at the store. Commercial means from Wonder Bread to artisan bread, but either way, you know what you're getting and you pay accordingly. But once you get home, you can use that ready-to-use bread and be eating your sandwich in 5 minutes. So both types of software enable you to share your sandwich and feed a friend if you like, mixing my metaphors already. Both sandwiches, I'm sure, are delicious, and both sandwiches, once shared, might inspire your friend to riff on your perfect sandwich and create their own. They're both good. And so what our goal for Neurolucida 360 is to be the best sliced bread that we can provide for you. We want to have the needed functionality and the needed capability that you need in order to drive your software or your experiment further, faster, and more complete. And our goal is to do that in a way that allows you to continue to do the creative process that you need to do to develop your experimental questions to their fullest. Today, we're going to talk about 3 topics, and we'll get into that in just a minute. But the 3 topics are where Neurolucida 360 is right now and where not only I would like to take it, but importantly, where you would like to see it go. Then we also want to spend some time talking about FAIR data and what that means to us as part of our initiative towards open science, and some of the ways that we are actually working towards the open science perspective here at MBF Bioscience. So I'm really glad that you've joined us today, and we're ready to dive into the details. Aidan, would you like to cover today's agenda?
Sure thing. So like Sue said, we're covering 3 topics today. The first is what's new with Neurolucida 360, and Sue's going to demonstrate some of the new tools that have been developed this year and talk about what's coming up for the future as well. And then she's going to hand the conversation over to me, and I'm going to talk about the new FAIR data tools available in NL360, as well as MBF Bioscience's publicly available file specification. And we will have a Q&A session following each portion of the presentation where you guys can sort of contribute to the conversation. And to submit questions today, you can do so simply by clicking on the orange arrow button in the GoToWebinar control panel. And that will open up your control panel where you can find the question panel. Type your question in the box and click send. And if you prefer, you can undock the question box from the panel for a better view of the questions and answers.
Thanks, Aidan. So today I'm going to start off by giving a little overview of some of the newest features in Neurolucida 360, a quick demonstration so that we can get into the meat and potatoes of what we would like to see in the coming year. So I'm just going to switch over to Neurolucida 360. And the imaging engine, so Neurolucida 360 has undergone a tremendous amount of development work over the past couple of years, and culminating with this most current release, 2020.3.3, that includes an overhaul of many of the engines that drive the functions of Neurolucida 360. So we have a data core, and we also have an imaging core that work together in order to create the data faster and more smoothly than ever before. So I have the 3D window open. I'm just going to make it a little bit smaller so it can fit nicely on our screen. And I'm going to load an image. And one of the first things you'll notice is how quickly the volumes load, not only in the 3D window, but also in the 2D window. We go to the image tool panel and select adjust. We can make adjustments to the image adjustment on a per channel basis, and this is instantaneously reflected in the 3D window as well. So that's a big change from what was previously possible. And what that means for you is that your data is easier to manage and easier to work with than ever before. On the left-hand side of the 3D panel, we have the channel panel. And so right now we're displaying all channels. This is a 2-channel image. And so if I select the green color swatch, you can see that the red channel is hidden. And if I select the red channel, you can see that the green channel is hidden. Now this just makes things easier to work with, but it also allows us to do some more sophisticated operations, such as assign trace data to individual channels. So what I'm going to do is I'm just going to automatically trace this axon here. And the trace data will be associated with the image channel. So now we know that the red channel has the axon tracing, and the green channel has nothing. And so I automatically trace this. Here's the tracing that results from just running voxel scooping. And so now when I go to the green channel, so I'm going to turn off the red channel and turn on the green channel, you can see that my trace segment is there, but it's the opacity is much lower, and you can't see the red channel at all. We're just showing the interaction here. And so this allows you to have greater flexibility and greater knowledge about what components of the data come from which features of the image data itself. And so this allows greater power for you to do enhanced analyses, as I'll demonstrate in just a second. So right now we have a axon, and all of these green specks here are V-glute puncta. So, so now I want to show off the puncta tool. This is brand new with the latest version of Neurolucida 360. It replaces the synapse detection. And so I'll just walk you through it briefly. So on the left hand or the right hand side of the panel, there are 2 methods for detection. You have an automatic detection, and then you have an automatic detection enhanced with machine learning. And so if I select the radio button for machine learning, that means that it will apply the classifier that we've already prepared and baked into Neurolucida 360 into detecting these punctate objects. And so we can get in here and click to detect, just like all the other features of Neurolucida 360. So we can get a feel for what type of detection we would get. But if we preview seeds, and then detect all, when you preview seeds, the machine learning takes a little bit longer than the automatic method, because we are running the seed detection through an additional criteria to make sure that it meets the training criteria for the machine learning classifiers. And so that will populate. And then one other thing that I would like to show you is this ability to detect based on proximity. So my first step in this process was to trace this axon And if I only wanted to detect the puncta that were within a certain proximity to the axon, I would check this box here. Okay, so now if we hit detect all, the proximity or the seed detection is going to happen, and all of those different modules, the puncta, are going to be identified and detected based on the classifier that we trained previously. But we have a lot planned for the application and incorporation of machine learning in the coming year. This is the first application that has been augmented with machine learning, and the process of training the classifier is based on the selection and identification of punctate objects that can be as small as presynaptic terminals or postsynaptic terminals to somas, any round object like perspective. And the goal is to continue to add additionally trained classifiers, as well as additionally applications of AI to demonstrate enhanced detection capability based on the image data itself. And so the process has completed, and now you can see just how many puncta were detected. And you can see that the less spherical objects were not included based on how we trained the classifier. So there's some size constraints as well as some shape constraints that were trained into the algorithm at that time to enable us to have a more discrete methodology for discriminating punctate objects, in this case, V-glute terminals in these structures. And so this is the puncta detection. And now you can see that if we show all of the channels, you can see the axon traced in yellow, and each individual punctate object identified. There's 1,241 in this particular volume. And then the number of analyses that you can do by simply selecting the button to send to Neurolucida Explorer is straightforward and easy to use. I'd like to open this up for questions. Does anybody have any questions about some of the new features about Neurolucida 360, or particular areas that you would like to explore in the coming year, or you'd like to make me aware of, so that I know to allocate enough effort in our development pipeline to drive Neurolucida 360's development in a perspective that would be most beneficial to you. I'm particularly interested in applying these machine learning principles to spine detection, because spines are gloriously relevant in image analysis, and I also think that they represent a complex problem that could be enhanced through training the algorithm to use enhanced criteria to identify the best shape and perspective for the dendritic spines. And I'm just going to expand my question window to see if I can see. Aha. So how long asked what is the puncta staining? So this is V-glute 1, and so it can automatically identify those particular objects by using the shape criteria that we had already determined. If you instead choose the puncta detection without the machine learning, one, it'll operate faster because it doesn't have to predetermine whether or not the seeds meet the criteria to be included in the sample. They use simpler criteria, such as detection intensity and size of objects. But you can augment that information using the machine learning if you so choose. So it's sort of a, your mileage may vary depending on how similar your image data is to the data that we used to train the classifier, but I will say that it seems to be pretty robust to multiple different formats, especially sizes. So it seems to work pretty well so far. The axon itself, James McKenna is asking whether or not the axon was labeled. It was filled after recording, so it was a recorded cell that had been filled. Andrew is asking whether or not machine learning can be trained by the user. Currently, the answer is no. And that's something that I would like to really open it up for to hear your thoughts on as a group. We know from our experience and research with machine learning that the process of training a classifier is, one, tedious, and two, time intensive. And it does benefit from a wide variety of image data in order to make the classifier as broadly based and specific as you need it to be. And so our current thinking is that we will continue to add classifiers to the software that have been trained by us, and we might open up, say a call to say we're looking for image data to enhance the next algorithm. Would anyone like to produce that or provide that to us to help train? The flip side is making it available for you to do the training. So I'd be very curious to know if that's something that you feel very strongly about, or if you would be happy if it was just plug and play for you. There is another question about training the AI for synapse detection. Does it depend on the quality of images, and would it matter, including the depth of the images as well as z distances, which will be used to train the classifier? And the short answer is, of course, yes. The quality of the image data does have an important consideration when you are identifying the positively labeled as well as the negatively labeled structures that will be identified for using to train the algorithm. And we have taken pains to make sure that we don't use crystal clean image data like this one only. And so we use a broad variety of image data. And I believe we trained this algorithm on over 2,000 objects across various different imaging sets. And that's a very important criteria for the process of training. I'm going to switch gears really briefly, and then I'm going to hand it over to Aidan. There is a question about detecting spines, and so I just want to show real briefly how the, oops. Actually, maybe if there's time, I'll get back to that. So if we have time at the end, I'll cover all the spine questions, because there's a bunch of spine questions. And we would especially enjoy having a little bit of time to talk about that and where you might want us to go in the future. Really, thank you so much for all of the questions that you've asked so far, and we'll circle back to them throughout the presentation. But I think I'm past due, and I need to hand this back over to Aidan.
Thanks, Sue. I am going to launch a question before I get started.
So, Aidan, I think this is a really important question. You know, whether or not people, there is a big drive in open science to not only create great data, but also share that so that it enters the public domain for other people to build upon. So not just your own findings, but your source data as well. And so there's more and more interest in the data repo side of things, and it would be great to know if you, as a group, are interested in contributing your data to data repositories. I know that one project it's required.
So it looks like we have a few that are planning to in the future, and some people that do currently share their data with a data repository. So that's great. This next topic that I'm going to cover is going to be very relevant for those who are sharing data to a data repository. So what I'm going to talk about next is a new initiative that MBF Bioscience is working on, inspired by the FAIR data principles. And the FAIR acronym stands for findable, accessible, interoperable, and reusable. And we are working on implementing this initiative within Neurolucida 360 currently. So basically, our focus is to make it easier for you to create data that can be reused by you, shared with your colleagues, and repurposed by others. The mechanism we've created works under the hood, so data you generate in Neurolucida 360 will have the necessary FAIR aspects with little effort on your part. These FAIR aspects include metadata about your sample and the structures modeled using Neurolucida 360's toolset. This metadata allows the morphological models to be interpreted at the file level, independent of you. MBF Bioscience makes the morphological model data more findable through a connection with SciCrunch, a terminology database that provides researchers with a dynamic organ-specific lexicon of biomedical terms to use while they're segmenting microscopy image data. The connection allows users to incorporate rich metadata with explicit data IDs that are searchable in the registry. The dataset provides a restricted set of terms that are presented to the Neurolucida 360 user within this software. It handles synonyms, abbreviations for anatomical terms, taking the guesswork out of interpreting the annotated structures. If we take a look at the slide here, screenshot 1 shows the Spark dialog right here. And that opens in Neurolucida 360 as part of this new FAIR toolset. And here you can enter information about the subject that the microscopy image originated from, including species, subject ID, sex, and age. And here I've input mouse for the species, I've added my subject ID, selected male for the sex, and for the age, I've selected 4 weeks. And what's useful about this tool is that you have the ability to mark sex and age as blind to condition in case that is a experimental control. Blinding the sex and age is an experimental control for your annotator. And then once your annotation is complete, you can go back and add that information into the data file. Over on the right side here, anatomical terminology lists that you would like to annotate your image with can be selected. These lists have been curated by anatomical experts and are provided through the API connection with the SciCrunch database, as I mentioned before. Here I've selected the mouse heart list, and this means that all of the heart terms for the list are going to be returned to the user in Neurolucida 360. So over in screenshot 2 here, you can see the brief first portion of the heart list here, as it would be displayed within the software. So you can see terms like the absent left coronary artery and the absent right coronary artery and so on. By selecting the corresponding term from this list, users can annotate that region within their microscopy image data. This technique was used in the example below. So let me talk about that for a minute. This experiment was performed by doctors Jack Chang, James Schwaber, and Raj Vadajpalli. Their labs are at the Thomas Jefferson University and the University of Central Florida. And these researchers used the heart terminology list to serially delineate anatomical regions such as the endothelium of the right and left ventricles, aorta, pulmonary arteries and veins, and so on. They then identified and counted intrinsic cardiac neurons, which are shown with the yellow markers here. The 2D serial outlines of the anatomical regions were used to generate this three-dimensional model of these heart regions, which you can see in the screenshot here. Now let's take a look at the information that's stored within the data files using this new FAIR tool. Screenshot 3 shows the Spark data section of the MBF data file. You can see fields like the subject ID, sex, age, and those terms are as expected, how you inputted them in the Spark data dialog up here. Oops. This is not true for the species field. You can see the species field here actually has an explicit ID as the value here. This ID is unique and searchable in a registry. And the same is true for the root ID. It has a unique ID indicating the terminology list that was selected for annotation up here in the Spark dialog. In screenshot 4, you can see an example of a contour element within the MBF data file. This contour is named the absent right coronary artery. And the trace association property within the contour, right here, has that unique identifier as well. And it indicates the specific anatomy term that was used to annotate that region within your data file. These terminology lists can be used to annotate a number of Neurolucida 360's data elements, including contours, which we described here, but also markers, trees, and vessels can be used. When these new features are released, it will be easy to produce fair morphological models of your microscopy data. With this amendment to the MBF Bioscience data file, this data type will have the ability to stand alone and tell its own story. The subject metadata provides a robust understanding of how trace data was derived. By embedding these unique anatomical identifiers within the corresponding data elements, people looking to repurpose this data know exactly what the element represents. These features have been developed through MBF Bioscience's participation in the NIH Common Fund SPARC, or Stimulating Peripheral Activity to Relieve Conditions, and they are not currently available to the general Neurolucida 360 users. But we think that this is relevant to all researchers, and we plan to make it broadly available in the next release of the software. So get excited about that. You may be wondering why I just showed you the jargon that is stored within the MBF Bioscience data file and why you should care about that. Well, I have an exciting announcement to make. MBF Bioscience has fully documented their file format for open use. And I would like to launch another poll question for you guys today. And this question says, Would you like to create your own analysis tools for the data generated using Neurolucida 360? So go ahead and respond with the answer that applies to you here.
Smiljana asks, what countries can take part in FAIR metadata data collection? And the short answer is every scientist can work to make their data more fair, so findable, accessible, interoperable, and reusable. The tools that Aidan just presented just now are our mechanisms to try to make that process easier for you to do at the time of data collection. We think that the best way to make it easier to collect FAIR data is to incorporate these principles at the time that you're actually sitting down to do image analysis. And so these tools that we have created for the Spark program, we're going to enable you to turn on in your own version of Neurolucida 360 or any other MBF Bioscience product. And you can add these metadata fields right to your data files in simple and quick steps. And we think that that will be available in the next release.
Thanks, Sue. So I want to share the results of that poll. Really exciting. About 50% of you said yes, you're interested in creating your own analysis tools from the data generated in Neurolucida 360. So that's great. You're going to find this next section about the public MBF file specification really interesting. That's great to hear. So let's move to the next slide. The file specification details each element of the MBF XML data file in an effort to encourage the reuse of these rich data files for alternative analysis or reproduction of derived conclusions. This specification is already used by developers that incorporate the neuromorphological specification in their workflows. We hope that this is a valuable resource for your team as well. And you can check it out at mbfbioscience.com/file specification. You can ask us any questions about the file specification on this web form here. And lastly, we have published a preprint version of our related manuscript on bioarchives. That link is also on the screen there. So take a look and discover what makes our file format for neuroanatomical structure modeling so impactful. I want to show you an example of a tool developer that is currently making use of the file specification in their tool development workflow. As a part of the Spark project, the Auckland Bioengineering Institute is able to use the MBF data file due to its unmatched FAIR data, computer-readable elements, and scaled coordinate space. You probably recognize the data shown on this slide from one of my previous slides. The workflow here demonstrates the experimental process performed by the labs of Dr. Chang, Schwaber, and Vadajpalli at the TJU and UCF universities. It starts with the imaging of the sample, which is then segmented using those FAIR anatomical terminologies that are available in MBF Bioscience software. In the middle here, you can see the final 3D product of the image segmentation. This data is written to the MBF XML file format and was shared with the Auckland Bioengineering Institute, or ABI, who build 3D finite element-based maps of internal organs. These are called scaffolds. You can see an example of the heart scaffold here on the right. Neuronal pathways segmented can be mapped onto their computed locations on the generic organ scaffolds, and that individualizes them and allows for comparison within and across studies. The MBF data file makes it easy to compute anatomical regions and fiducial locations used for fitting the subject-specific segmentation data to these organ scaffolds. Additionally, the open file specification allows ABI developers to incorporate the necessary elements of the MBF data file into their tool development workflow. Other users of the MBF data file include data repositories like Spark, the Blue Brain Project, the Human Brain Project, neuromorpho.org, and additionally, software tools like Neuron and Biolucida can also read the MBF data files. You may recognize the Biolucida is another MBF Bioscience product, and it's traditionally used for visualizing microscopy images over the web, but it is now able to read those MBF data files into its Webviewer as well. So let's go ahead and take a look at that. I'm going to demonstrate how to use that data viewer. So here on the MBF Bioscience website is actually an embedded iframe of the new data viewer tool. So you can see that this example of one of the MBF data files is right on our web page here. And you can just zoom and rotate, and you can also use the Shift key to click and drag to move that tracing within your screen here. So you can fully explore all of these data files that are up on the website right now yourself. So I encourage you guys to go take a look at this. It's pretty cool.
And we're going to send you a link in after the conclusion of our meeting today, in case you want to be able to find this and play with it yourself. But this gives you an idea of what types of elements can be identified and mapped using Neurolucida 360, and also some of the complexity of these cells that have been identified and mapped by users of our software.
Thanks, Sue. All right, let me jump back to our slides here. And just as Sue said, you can try out the new data web viewer at mbfbioscience.com. And here's the link to get there. Additionally, go ahead and explore that file specification. Or if you have any questions following the presentation, you can contact us directly at info@mbfbioscience.com. So just send us an email if any questions come up later on. But that concludes our presentation today. Thank you guys so much for joining us. It was great to have you and to get your feedback through those polls and questions. But I would like to open it up to the question portion of the webinar. So if you have any additional questions for either Sue or I, please go ahead and put those into the GoToWebinar question panel, and we will take some time to answer those.
I know that I'm really excited about the steps that open science principles have led to such a dramatic proliferation of new tools, new interactions, and new collaborations that I've seen. And I'm hoping that the steps that MBF are taking makes it easier for people who use our products or would like to use data that has been created in our products to go the next step further. So whether or not you want to take the puncta detections that of a file that I just demonstrated, and then write a MATLAB script in order to do some complex colocalization analyses and factor that into, say, your optogenetic in vivo imaging, that may be something that is a highly specific question that you have, and you need to script that in order to get that information. But we want to make it easy for you to use parts of the pipeline that we can help you with. And I'm hopeful that the documentation of the file spec and the components that we're putting in to make it easier to integrate additional experimental details about your experiment to the segmentation file is something that just makes that process travel more easily, so that if you come back to your experiment 10 years from now, you'll be able to know what the age of that subject was, the sex, and what region of the brain you were looking at when that beautiful pyramidal cell was reconstructed. Those are things that I think are really pretty cool. Hey, Aidan, would you mind giving me control back? There's a few questions about spines, and I would love to talk about spines. Dendritic spines are just gorgeous.
Sure thing.
Okay, so I have Neurolucida 360 up, and what I thought I would do is this is a simple data file, or a simple image file. This is image data of a particular apical dendrite imaged approximately 100 microns from the soma in the hippocampus. And what the experimenters were interested in understanding is, for this particular dedicated region from the soma, how do dendritic spines change in their experimental paradigm? And so that's why it's a simple segment, and the image data was deconvolved after acquisition. So these cells were filled and then imaged. And so to demonstrate dendritic spine detection and classification, the first thing we need to do is trace the dendritic branch, because dendritic spine detection is relevant to the dendrite itself, because some of those parameters that we're going to get into about spine classification are all based on the dendrite itself. So how big the spine is from the surface of the spine dendrite, as well as other components. And so I've got a dendrite trace here. And if we go to the spine panel, you can see that there are detection settings here. They're all completely customizable. The detect all will use these parameters in your image data to model the individual spines. So you can do what I call click to detect, where you can select an individual object and have the algorithms automatically detect it. Or we can identify individual parameters. This is a really clean image, and many images have either more shot noise due to the imaging device that you were using, or there may be, if you're using Golgi, you've got a lot more components, especially Z-axis smear due to the bright field imaging modality, that can have an impact on spine detection. And so we know these things, and we've incorporated the different imaging parameters and capabilities into the detection algorithm to accommodate the differences and the impacts of image resolution on your ability to detect spines. And so what we do is we break this up into two components. There's the idea of detecting the objects, so the dendritic spines, and then there's this secondary effort for classification. And so for classification, what we're going to do is using the shapes of the objects that were detected, apply classification rules to them. And so we can automatically classify these dendritic spines into 4 categories: mushroom, stubby, thin, and filopodia. And you are in complete control over what the classification settings are used. And so you can dial in a head to neck ratio that is different than 1.1, as well as the length to head ratio. the size of the head is important for the distinguishing characteristics between mushroom and filopodia, and we know that those differ not only in development but also in brain region. And so we wanted to make sure that we have this mechanism that is flexible enough for you to modify these parameters as needed in order to suit your particular perspective. And so if we return to classify these guys, you can see that the vast majority of these spines actually ended up getting classified as thin because they're the head diameter of the dendritic spine is not greater than 0.25, or I think it was 0.25, I can't remember, but the cutoff for the mushroom head. And so you can select an individual spine, see what type it is, and you can also, if you want to have a better look at it, oops, green is already, you can change the color, and so that when we look at Neurolucida Explorer, you can see that the vast number of analyses that are possible within, oops, I hit the wrong button, are possible within automatically included in Neurolucida Explorer. So I'll give this a name, save it as our as an XML file. And Neurolucida Explorer will load this data file. You can see all the different dendritic spines. And now what you can do is you can easily pick out that one that you wanted to get a better look at. And so if you are interested in using your own classification parameters, this is an excellent way to get a feel for how to bin your parameters by being able to say, I'm going to manually classify a number of these spines and see what the range of physical sizes are so that you can create those detection or the classification parameters that suit your needs. And so if we go to analyze, we can do branch structure analysis and spine details. And you can see that we have, oops. Out of all of the spines that were classified, you can see that 68 were thin. Oh, actually, I made this window much too big. But we can see that you can just select interactively within the window and it'll highlight these individual data for you. And so you can see that this aqua colored spine that I recolored is still classified as a thin spine. If I wanted to change that in Neurolucida 360, I could. And all of these types of detection parameters can be accommodated and adjusted for you as you go through and do your detections in Neurolucida 360. I'm just going to check back at the questions and see if that helped to address any of the questions that Gaurav and Pooja mentioned about dendritic spines and their interest in dendritic spines. If you have additional questions, just let me know.
So Gaurav did have one more question that I can help answer. He asked, in order to model the dendrite, which algorithm suits best? Reverse crawl, voxel scooping, or oh, I'm blanking on the last one.
Directional kernels.
Thank you. Directional kernels. So that's really going to be image dependent, Gaurav. And we provide all three of those algorithms to the user there to sort of, as you load in your image, you can choose one of those and try it out on your image and determine which algorithm is going to work best for the image that you have in particular. Most of the time for the staining that we use, for the stainings that we get in for dendrites and such, I would suggest directional kernels personally, but the other ones are there for you too in case they do work better on your specific image data.
Yeah, so I'll just give a little bit of insight into how to choose which algorithm to try first. So directional kernels, so a key component of all these algorithms is to determine foreground from background, right? You need to determine what is the object you're trying to segment from the background in order to start segmenting. And directional kernels looks for the external edge of, the sharpest line between dark and light is how the foreground is determined for directional kernels. So that is really good for discriminating these edges, especially in cases for bright field data and for discrete punctate labeling. Voxel scooping and Reber's crawl both use an inside out, so a region growing methodology for tracing the dendritic branch. And so it's going to start at the brightest voxel and continue to build the dendrite moving out until it reaches that cutoff between foreground and background. So an inside out mechanism is great for filled cells, for really densely labeled structures. So if you're looking at something that is intensely complicated with lots of branches in close proximity, I would recommend starting first with voxel scooping or Reber's crawl. If you have a filled cell with, say, biocytin, where you get these breaks or incomplete fills, I would recommend directional kernels for that particular perspective.
Well, it looks like we are coming up to the end of our hour, Sue.
That went fast.
Well, thank you again for joining, and I wanted to just let you all know that if we didn't get to your question today, we'll be sure to reach out to you following the presentation. So don't worry, we'll get to those questions. And we hope to see you guys at the next SfN socials coming up for MBF Bioscience as well. And thank you again for coming.
Thanks, everybody. Enjoy the rest of the SfN Connectome.